Okay, we have 10 students now. I have started to record today's class just in case, if they don't come, they feel that the class is canceled, they will still be able to view the content later on. So today we're going to discuss the principles of stem cell biology. So first of all, we want to discuss what is a stem cell. So I want you guys to provide me your impression. What do you think is a stem cell? So you can unmute yourself and volunteer to speak up. Tell me, what do you feel the definition of stem cell should be?
A
So
stem cell should be it's a cell that is undifferentiated. It hasn't committed to a lineage.
Great, great. Anything to add?
a type of cell that can turn into like any kind of cells you want it to be. Like you can grow any organs.
Okay.
Out like it's just like a basic parent cell, and you can turn it into like it's kind of like clay. You can like water it, and you can like make it to whatever you want.
Yes, yes. Appreciate that. Yeah, you I will correct you because we're trying to provide the definition of stem cell, but you both of you said the one of the most important property of stem cell, but there was something that's not entirely correct. Later on, we're gonna clarify that. Anything to add? Okay, if not, I'll give you the
Oh, I
definition.
was gonna add something if that's okay.
Yes, you go ahead.
So they're self-renewing. So
Yeah.
they gotta you can't let them deplete because that'll be a serious problem that can lead to diseases. So, yeah, I think that's pretty. And
Absolutely,
yeah.
absolutely. With three of you guys provide your impression about stem cell. Now we have the definition of stem cell. Uh, what's going on? Let me see, where is it? Okay, I have to click on the or on my screen for it to appear. So a stem cell is a special kind of cell that has a unique capacity to self-renew, to renew itself and to give rise to specialized cell types. So basically, we're, we, it must have two criteria. One, self-renewal, the other is differentiation. So the first two students focused on the the criteria of differentiation. So basically, stem cell, by the name stem, that means they haven't committed to anything yet. So later on, once they start to differentiate, they will become specified cell, and then they can perform a defined function. So both of the first two students focused on that part. The stem cell must be able to give rise to a specialized cell type. And then, the third student, I'm sorry, I will call your name later once I get more and more familiar with everybody. So he added that stem cell must have the ability to self-renew. That is the other very important property of stem cell, because if the stem cell cannot self-renew, and then they will just go down the road of differentiation and get themselves depleted, which is not desired. The stem cell is a pool of cells that's reserved there to meet certain circumstances, such as injury, for example. Like when you have a skin injury, your stem cell will become activated, and then they will differentiate and replace the damaged tissue, right? So in the future, when somebody mention a particular cell type to you and ask you if that is a stem cell, remember you have these two criteria. They just try to determine whether that cell type meet these two criteria. So look at that cartoon shown on this slide. The stem cell, although they have two properties, one self-renewal and the other is differentiation, it doesn't mean they are doing that all the time. They can be quiescent. That means they go out of cell cycle. They just they will hide somewhere, keep a minimal activity and metabolism. They just stay there, keep their identity. But once the environment or circumstance ask them to behave like a stem cell, they're going to go out to either self-renew or start to differentiate, right? That doesn't mean a stem cell must proliferate. They must either self-renew or differentiation. It's not like that. So they can be a quiescent cell. And then when they differentiate, when they start to differentiate, usually it also involves cell division. So oftentimes they go through a process called asymmetrical cell division. That means when they divide into two cells, one of the cell will be identical to the stem cell itself. So they create one stem cell and another cell, which is often a progenitor cell. So they often go through an asymmetrical cell division, creating two cells that are not identical to each other. That's why it's called asymmetric. But sometimes they do go through symmetric division. In that occasion, oftentimes they create two identical stem cells. That means, that process doesn't involve any differentiation. It's just a cell division creating two stem cells. And extremely rarely, they can become two progenitor cells, which has a cost, because now you are depleting the stem cell pool. Only when when the situation is very, very bad, like a big injury, maybe some of the stem cells will have to be depleted, creating two progenitor cells and go down the road of differentiation. But most times, they do self-renew and differentiation at the same time. They create a stem cell, which is the self-renewal part, and then they create a progenitor cell, that is the differentiation part.
Dr. Liu,
yes.
Question. So I was wondering, do we know what regulates between asymmetric and symmetric division? Like, how do they know which one to choose?
Well, that really depends on the situation.
Okay.
So if they, for example, like there is a damage, you need the circumstance require some cell replacement, and now probably it's going to go through a asymmetric division. Okay. But regularly they are either a quiescent cell. If they did split, they split to two stem cells.
Okay. All right. Thank you.
Yeah.
I have a question.
Yes.
So do they go through meiosis and mitosis?
So they go through mitosis. It's not meiosis.
Okay.
It's not meiosis. So the germ cells are not actually stem cells. They're just a somatic cell, a very specific somatic. No, they are germ cells, not somatic cells. They're germ cells, but they perform a specific function. They are not stem cell. There are stem cells for germ cells. Some particular stem cells that can give rise to germ cells, but germ cells are terminally differentiated cell. So, germ cells go through meiosis, while other cells go through mitosis. Stem cell goes through mitosis, not meiosis. Okay, so here, this cartoon illustrates how stem cell go down the differentiation road. So you see a asymmetric cell division, like in the previous slide, the stem cell now become one stem cell and one progenitor cell after the asymmetric cell division. But usually, this progenitor cell will go through a phase of proliferation. So they will expand themselves. Therefore, this stage is sometimes called a transient transit amplification. The cells are called transit amplifying cells. So you can say that the progenitor cell serves the purpose of amplifying the stem cell. So this is at the early stage of differentiation. Stem cell become progenitor, and the progenitor will amplify themselves by proliferation. And after multiple steps, eventually they become the terminally differentiated cell type. Okay, so it is very important to distinguish that stem cell and progenitor cell are different. So it's very, very easily confused, these two identity. Because, you know, if you have, for example, in the embryo, some cell during embryogenesis, they will eventually become the heart muscle cell, but they're highly proliferative, but they don't perform any function. They're highly proliferative, and eventually they will become heart cell. You will feel like, well, that's probably a stem cell because they're proliferative, right? And then they differentiate toward the terminally differentiated cell type. You feel that is the stem cell, but you have to try to figure if that particular cell type fit the two criteria. For a cell to become stem cell, it must be able to self-renew. For progenitor cells, even if they are proliferative, they can divide it into 2 cells and then 4, and then eventually get differentiated. They cannot renew themselves. That means during those proliferation, each cell division will make a different cell type, slightly different, right? So they're not creating themselves. They are not doing self-renewal. Each cell division, they're creating a slightly more specified cell type. Although it's not yet terminally differentiated, but it's a differentiation process. It's many, many steps. So you look at the cartoon, you see that the state of differentiation gets greater and greater along the proliferation and differentiation, right? So when the cells still are proliferative, but during that process, you're gaining specification over time. At the same time, usually their proliferative potential are dropping. So at the beginning, they're highly proliferative. They grow really, really fast, but gradually they grow slower and slower. At the end, they don't proliferate at all when they become the terminally differentiated cell, like your heart cell, the neuron cell in your brain. They don't proliferate it at all. Okay, so remember, this is a long process. Cell differentiation is a long process. At the beginning, usually the cells are proliferative, but gradually they are not growing as fast as at the very beginning. But when the cell proliferative, but they lose their stem cell property, they're no longer stem cells. Okay, so try to distinguish stem cell and progenitor cell. The progenitor cell are proliferative, but they're not self-renewable. They're not renewing themselves. Okay, that's a very, very important concept there. And based on the origin of detection or the origin of isolation, we can divide the stem cells to 2 types, 2 categories. One is stem cells in the embryo. The other is the stem cells in adult tissues. But I usually don't try to say the cells that you identified from embryo is embryonic stem cell, because embryonic stem cell is a special term. Later, you're going to learn it. When you say a cell is embryonic stem cell, it's a very specific cell identity. But in the embryo, you actually can isolate quite a few types of stem cells. Therefore, I don't say that the stem cells in the embryo is embryonic stem cell. I hope this is not confusing, because that term, ESC, is a special term you use to only refer to one of the cell type, one of the stem cell type in embryos, where you still have other stem cell types in developing embryo. Okay, but it's not confusing when you refer some stem cell as adult stem cell. That means, they are in adult tissues, like in the brain, like in bone marrow, even in the heart, you can identify some stem cells. So with that, I also want to clarify one concept, because the second student who tried to give his impression of stem cell said that stem cell can differentiate into almost any cell type that you want. That is actually a confusion between stem cell and embryonic stem cell or pluripotent stem cell, because some stem cell, like embryonic stem cell, does has the potential to go to any cell types, almost any cell types you want them to become. They're highly flexible. That's the identity of them is highly flexible. You can give them the correct cue and then lead them into any cell type you want. But a lot of other types, like adult stem cell, they have limited differentiation potential. So, for example, like the stem cell in the brain may be able to differentiate into 2 or 3 cell types. They absolutely won't differentiate into heart muscle cells. They won't differentiate it into blood cells, right? So their differentiation potential is limited. So I hope that's not very confusing. On this slide, what I'm trying to describe is that stem cells can be detected or isolated from either embryo or from adult tissues. And often, the stem cells in embryo have higher potency. Well, the stem cells in adult tissues have limited potency. So that is, the potency of stem cells. But on this slide, I want to point to you what is embryonic stem cell. I said, there could be many stem cell types in developing embryo, but only one stem cell is called embryonic stem cell. I want to describe what is that. What is embryonic stem cell? So here on this slide, it shows how embryonic stem cell is derived. So basically, this has to do with early development. So let me briefly, describe what's happening. So usually you have fertilization. The egg will receive the sperm, like at the very beginning. You have the fertilization, which means the sperm penetrates the cell membrane of that egg and go into the cytoplasm of that egg. That's fertilization. Then that is called a fertilized oocyte. And then it goes through a process called cleavage. During cleavage, you don't see a change of the size of that embryo or the oocyte. Oocyte is very, very big. It's a big cell. It's the biggest cell in our body. And during cleavage, you have many, many types of cell division, but you don't have a net increase of the embryo size. That means through those cell division, you're creating smaller and smaller cells. I hope that's easy to understand. You have a big ball, that's one cell, and then through cleavage, you're making that big ball to become many cells. The net size of that big ball is not changing, so the cell number is growing, but the cell size will become smaller and smaller, right? At one point, the embryo will have 8 cells. The 8 cell will mean that you had experienced 4 times of cell. 1 time create 2, 2 times create 4, 3 times create 8 cells. So after 3 cell division, and now you have 8 cells. That's an 8 cell embryo. At that stage, all those cells, all that 8 cells, theoretically can give rise to any cell types in our body, including the trophectoderm, which is, the germ layer for placenta. Okay, only before the 8 cell embryo stage, the cells can give rise to the embryo proper as well as the extraembryo proper. After that, the 8 cell embryo will develop into a structure called blastocyst. So the blastocyst have this structure with a cell mass at one end of the structure. So that cell mass is called inner cell mass. The inner cell mass, if you take them out, just this part, the inner cell mass, if you take them out, this can grow as a cell line. And now you call those cells embryonic stem cells. That's how you derive embryonic stem cell. The embryonic stem cell is from the inner cell mass of the blastocyst. But here, there is a change of the potency of the cells. As I said at the very beginning, if you isolated the cell before the 8 cell embryo stage, all the cells can contribute to all the tissue types, including extraembryonic tissue types such as placenta. Okay, but in the stage of blastocyst, you get these cells, but these cells can no longer go to the lineage of extraembryonic tissues such as the placenta. So you lose some potency during development. Therefore, we say that the embryonic stem, I shouldn't say embryonic stem cell because it is a special term. The stem cell before the 8 cell embryo stage is totipotent. Toti means everything, all. They can go to everything. But embryonic stem cells, now it's a very special term referring to the cells derived from blastocyst. They can only differentiate into embryo proper. That doesn't include extraembryonic tissues such as placenta. These embryonic stem cells won't be able to contribute to the placenta. Okay, so here we have a major potency drop here. The totipotent cells give rise to everything. Pluripotent cell, practically, they can give rise to the things we want, except one cell type. That's extraembryonic tissue. They can't. Well, I mentioned already, the adult cells, adult stem cells will have a limited potency. Now, here is another look of what's shown already on the previous slide. So this is zygote. The zygote basically is the fertilized egg. You have 2 pronucleus. The nucleus here, nuclei, should I say, because once the original nucleus from the female, well, the other nucleus is from the sperm. But once the fertilization has completed, you don't say them nuclei any longer. You call them pronuclei. Pronuclei. That's the name for, just the term for the nuclei at this stage, pronuclei. So this is a zygote, and then the zygote will go through cleavage, giving rise to morula. And morula you can see there are 8 cells here. Either the zygote or the morula will have the ability to differentiate into anything you want, but morula later on will differentiate into blastocyst. You see inner cell mass there. This is called the inner cell mass. You take the inner cell mass out, they become embryonic stem cells. It's very special cell type, which is, I would say later on, we're going to describe pluripotent stem cell, and this is the gold standard of pluripotent stem cell. I have had my personal experience extracting embryonic stem cells. So you just mate the female and mate animals. Usually it's a rodent, mouse. You mate them, and after 3 days, precisely after 3.5 days, that's when that embryo will become a blastocyst. So usually we just go there, sacrifice the animal, take out the uterus, and then use a pipette to flush out, liquid to flush out the blastocyst. And then we put the blastocyst in culture. Those. Let me see. Okay, I can mute everybody, but it stopped. So the cells from blastocyst will migrate from blastocyst automatically. So they will become, the stem cells. And later on, you can scrap off the rest of the blastocyst. The inner cell mass will grow very, very rapidly. They are going to dominate the culture and become the embryonic stem cell line.
Professor?
Yes.
I have a quick question. So
Sure.
From the blastocyte, when we take out the inner, from the blastocyte, when we take out the inner cell mass, it will form the embryonic stem cell?
Yes.
Okay.
Yeah. So in practice, you don't have to take out the inner cell mass.
Mm-hmm.
You can take out the blastocyst, which is the embryo. The
Okay.
embryo at that stage is called blastocyst. You just take out the embryo and put the embryo on a culture dish. The inner cell mass will migrate out of the blastocyst and then start to grow.
Oh, okay, perfect.
Yeah.
Mm-hmm. So what would happen then to the blastocyst after the inner cell mass leaves?
They have other cells in there. They are also going to grow, but they grow not as rapidly as inner cell mass.
Oh.
So you can mechanically remove the blastocyst after the embryonic stem cell migrate out and has been established.
Okay, that's a nice trick.
Okay. Yeah, it was easy for me because some scientists have established that. It wasn't easy for them. And because of that, the simple operation that they have found, they won Nobel Prize.
Wow.
Part of reasons.
Yeah, makes sense.
Because of that. Okay, so there are 4 levels of potency. One is totipotent, the other is pluripotent, multipotent, and monopotent. I'm sorry that table is quite small, but if you get the book, you can. It's table 1.1 of that book. So I have explained already the difference between totipotency and pluripotency. That's extremely important because, practically we actually don't care about totipotency because totipotency is kind of like reproduction. It's kind of like cloning. It can be done naturally, right? We don't care about totipotency. The placenta, the trophectoderm are not extremely useful, but we are most times interested in pluripotent. We're interested in embryonic stem cells Yeah. So how to establish pluripotent stem cell is the task, for many years in the field of stem cell research. Like establishing embryonic stem cell, you're establishing pluripotent stem cells. So embryonic stem cell is the gold standard of pluripotent stem cell. Okay, the pluripotent stem cell can differentiate into anything except that trophoectoderm or placenta. But multipotent, you see that prefix pluri and multi, they're quite similar. Pluri means, everything except placenta, but multipotent is much more limited. Like in the previous slide, the brain stem cell can differentiate into 2 or 3 cell types. That's called multipotent. Okay, and monopotent is easy to understand. Although they are stem cell, but their ability to differentiate is only toward one cell type. The good example will be the satellite cell in your muscle. In your muscle, there is a stem cell called satellite cells. Satellite cell can only give rise to muscle and nothing else, give rise to skeletal muscle. So that they're only monopotent. Another example will be your skin stem cell. The skin stem cell will only replace your damaged skin, or over time, every day, some part of your skin will get replaced by stem cells. Okay, so those are important monopotent stem cells. So stem cells are different in their potency. So now you know that. So we can't say that a stem cell can give rise to everything. No, some stem cells can give rise to everything.
Professor, I have a question.
Yeah.
Are these slides on Blackboard?
I put it to Teams class materials.
Teams. Okay, thank you so much.
After today, I'm going to put it to Blackboard as well.
Okay, thank you.
But definitely get that book. This is a very, very good book. I've never seen a stem cell biology book as good as this one. It's a new book published last year, so it's worth buying, in my opinion. So I mentioned in the previous slide that we are mostly interested in pluripotent stem cell, including embryonic stem cell. On this slide, I want to tell you 3 ways to generate pluripotent stem cell. Of course, I have already described the primary way of generating pluripotent stem cell. So that is the natural way. So you make the animal, or in human, you can do in vitro fertilization or do it the natural way. So either way, as long as you can get the zygotes, the fertilized egg, you allow the zygote to develop into the blastocyst stage. Within the blastocyst, you can extract the inner cell mass. And now you can establish embryonic stem cell. This is the natural way to create a pluripotent stem cell. But later on, actually not later on, one of the approach was described even earlier than the finding of embryonic stem cell, which is called somatic cell nuclear transfer. So basically, there are 2 other ways. Both are artificial ways to generate pluripotent stem cell. So one is called somatic cell nuclear transfer. So in that method, what you do is you take the nucleus of a somatic cell. For example, the breast cell in the dolly shape, right? You can take a somatic cell nucleus, and then you take an egg and remove the nucleus from the egg. Okay, so in that egg, you don't have a nucleus because that nucleus is removed. It's mechanically removed. And now you put that somatic nucleus into the egg or oocyte. Amazingly, that structure can go through the natural development as well. it's amazing. And then, you can allow that structure, the somatic nuclear transfer engineered structure to go through embryogenesis toward the stage of blastocyst. And now you extract the inner cell mass, you can also create embryonic stem cell. So that is also called reprogramming. Why? Because that's the cytoplasm of that egg somehow reprogrammed that somatic cell nucleus. Because later on, you're going to learn the nucleus of a somatic cell and the nucleus of oocyte are very different. Although they have the same genetic material, but they're epigenetically different. That means they don't express the same genes, right? Later on, we're going to go into the details. So the somatic cell nucleus must be reprogrammed by the cytoplasm of the oocyte. Therefore, they go through normal differentiation or development. So that is one way to create pluripotent stem cell. And then about 15 years ago, this Japanese scientist, Shinya Yamanaka, had this major breakthrough. He found that he can put transcription factors, 4 transcription factors, into somatic cells and now get that somatic cell reprogrammed toward pluripotent stem cell. And in the third method, you don't even need an embryo. You don't even need an egg. You just put transcription factors into somatic cells, and after a long process, that cell will become a pluripotent stem cell, just like embryonic stem cell. But because this is reprogrammed by exogenous factors, so we call this induced pluripotent stem cell, IPS, induced pluripotent stem cell. But if you compare the property of ES, embryonic stem cell, or the somatic cell nuclear transfer engineered cell, or IPS cells, they are extremely close to each other in their identity. The differences are tiny. So there are 3 ways that we can use to create pluripotent stem cell. Shinya Yamanaka's work was great because not only he invented a way to create iPSCs or pluripotent stem cells, he also paved the way. He also had a conceptual breakthrough. And now we know that the cell identity is not fixed. It's not like the somatic cell will always be a somatic cell, the stem cell will always be a stem cell. The cell identity actually can be manipulated artificially. So that process is called reprogramming. Like described on the previous slide, you can make a somatic cell become a pluripotent stem cell by putting some transcription factors there. And later on, others described you can change one somatic cell into another type of somatic cell. Like you can change a heart muscle cell to become a maybe skeletal muscle cell or maybe blood cell. That is possible. You can make a lymphocyte to become, for example, skeletal muscle cell. Those are all now possible because the cell identity is flexible. You just need to come up with the ways to do the manipulation. So that is usually done by transcription factors, but not necessarily transcription factors. Other factors such as microRNAs, long non-coding RNAs, they can all be explored, to see if they will be able to convert cell from one identity to another identity. So it is amazing that some somatic cells can naturally transdifferentiate into another cell type. like in some types of injury in lower species, the one cell type can actually, become another cell type. So in nature, that happens. The cells can change their identity, but in a test tube, we can make that happen by doing reprogramming. So the cell identity is flexible. It's all because of the Shen Yamanaka's discovery. We now know that. But in their textbook, it's interesting that they mentioned that, to change the cell identity from one somatic cell to another somatic cell, or you change one type of adult stem cell to another type of adult stem cell, is not necessarily easily achieved. So what I'm trying to say is, if two somatic cell types are close enough to each other, for example, like the heart cell, the heart muscle cell, and the blood cell, maybe it is doable. You can reprogram the lymphocyte toward a muscle cell, which is doable. Why? Because originally they belong to the same germ layer. They're both from the mesoderm. But if you want to change one cell type to an unrelated germ layer, for example, like the brain cell usually is from the ectoderm, while the heart cell is from mesoderm. Now it becomes difficult, more difficult. Whether it is possible, I think it is highly debatable at this point. But I don't see that is reasonable, because if you can change a somatic cell to the stem cell, and why you can't theoretically, overcome the barrier, change from one germ layer to another germ layer. So theoretically, I think it is still possible, but practically it's not achieved yet. It's rarely achieved. So to change one somatic cell to another somatic cell type, at least for now, we're still trying to change the cells that are kind of closely related to each other. Okay, on this slide, I want to mention something that's very interesting. So in our body, different tissues vary greatly in the rate of cell replacement. And that may be a reflection of the number and function of adult stem cells in that particular tissue. For example, in our body, some tissues are highly proliferative. Like, I'm sorry, it's not highly proliferative. It's you have a high rate of cell replacement. For example, like skin and lining of the intestine. Here I have the number. So basically every 5 days, the entire lining of the intestine is replaced. There's a high rate of cell replacement. And of course, hematopoietic system, we always know you have high rate of cell replacement. And if you in some occasion, like in an injury, somebody lose a lot of blood, but eventually he's going to be fine. All the blood cells will be regenerated. So the cell replacement ability in some tissues is extremely high, but strangely, in some other tissues, you almost don't have any cell replacement, like in the brain and in the heart. My research field is in the heart. It's about heart development. It is very, very strange why the heart cell won't replace themselves. So I can have this statement here. So everybody born with certain number of heart muscle cells. And maybe after just 5 days, that number will never change again. So after 5 days after birth, so how many cells you have? It's going to be what you're going to have forever during your lifetime for your heart, which is very strange because the heart muscle works extremely hard. Every minute will beat 70 times. It's going to beat lifetime. it's hard to imagine you don't need to repair the cells, you don't need to replace them. So will you accumulate damage there after working so hard? So which is something interesting. So we still don't have a reason why, you know, some tissues have high rate of cell replacement, but some tissues just don't have that high capacity of cell replacement. I guess one of the reasons is the high rate of cell replacement often comes with cost, like the potential to have cancer, to have malignancy in those type of tissues. Like you see cancer in the blood system, of course, have all the leukemia, right? This malignancy of the hematopoietic system. You also have problems, skin cancers, things like that, but you rarely have heart cancer. Have you ever heard somebody have a cancer in the heart? Not really, right? Extremely, extremely rare. Probably that's one of the reasons, but still, it's you know, it's baffling. Then why we like to study stem cells? Here I list some benefits, healthcare benefits that we can have. Like, you know, we know that loss of the stem cell is not desired, which often leads to pathological conditions such as bone marrow failure, muscle wasting. So we are trying to understand, you know, what's the mechanism of stem cell loss? Can we prevent stem cell loss? So, you know, even if we get old, we're going to have the natural muscle wasting, but if we will figure out the mechanism of stem cell loss, maybe we can prevent that. So the stem cell can always replace the damaged muscle or other tissues, so we can stay functional for longer time. So that's one benefit. We study stem cell because we want to prevent the loss of stem cells. And another thing is it's cancer related, because we think that a lot of cancer is because of, you know, aberrant gain of stem cell properties by somatic cells. Some somatic cells now all of a sudden they gain the properties of stem cell. Well, if it is not a strict stem cell, they could become a related cell type, that's cancer cells. They can self-renew and then become a mass of different cells. Usually in a cancer, you know, it's not one cell type, but many cell types. Okay, so cancer is related to stem cell. So by studying stem cell mechanisms, maybe we can develop tools to give a cure to cancer. And of course, you know, the most important reason we study stem cell biology is because we can make stem cells to help repair damaged tissue. That is the topic of regenerative medicine. Also, you know, that's a topic that deals with the mechanisms of tissue and organ regeneration. So on this slide, I show some of the benefit. Again, at the bottom of this slide is tissue regeneration, which is self-explanatory. So I won't go there. You can always replace the damaged tissue with artificial source. You know, you can culture a cell and put into patient and help the tissue to recover. But additionally, on this slide, you have some other applications of stem cell. For example, like you can do toxicity testing. So you can culture the stem cell, make the stem cell to become a certain cell type. For example, like heart muscle cells. In a clinical, it's known that a big part of the drugs in development will fail because of cardiac toxicity. So they're going to cause arrhythmia. A lot of drug in development later will fail. They cannot go through the approval process by FDA because they're going to cause toxicity in heart muscle cells. And now, if you have abundant heart muscle cells, you can do the test. You know, don't have to go to clinical trials. You just test your drug on cultured cardiac muscle cells. Maybe you can start to see toxicity already. So building tissues by raising stem cells will give you that possibility. So I would say that the benefit of stem cell research is sort of endless. You know, you can always come up with other ideas why it is so important to study stem cell biology. But on this slide, what I want to explain is there are many paths, you know, that we can explore to use stem cells in clinical healthcare. So basically, both pluripotent stem cell and adult stem cell may be used for that purpose. So if you start with embryonic stem cell, which is pluripotent stem cell, you know, you can hopefully, you know, inject them into some patients, and then those stem cell will differentiate toward adult stem cell and eventually get fully differentiated toward the ultimate cell type. So you can start with a somatic cell and do either somatic cell nuclear transfer or do Yamanaka's iPSC generation. You first create pluripotent stem cell, and then you expect that this pluripotent stem cell eventually will give rise to the differentiated cell type, either in vitro, that means you make them to differentiate first in a test tube and then inject, or you inject your stem cell and allow the cells to differentiate in the body. So you can start with embryonic stem cell, or you start with somatic cell, but first get them to the stage of embryonic stem cell or pluripotent stem cell. So that is one path there. But I think most scientists and industry, they focus more on using adult stem cell. So similarly, they will expand and differentiate the adult stem cell either in vitro, that is also called ex vivo, that means outside of the body. You can make the cells to expand and differentiate and later do the injection. Or you can come up with ways, you know, to isolate the stem cell, or even without isolation. You know, you can come up with a drug that can activate a particular adult stem cell. For example, in your heart, there will be some stem cell. You come up with drug that can induce the differentiation of the adult stem cell. Previously, if a patient will have a heart attack, the stem cells are somehow not activated or not activated sufficiently. So the repairing is not sufficient. And now you come up with a drug. You can make those adult stem cells to expand and then make them to differentiate. So maybe the patient will benefit from your manipulation. So you're not taking the cells out of the body. You use drug to activate the stem cells. So here are just a few paths that maybe we can explore to use stem cells in regenerative medicine. So, you know, you can do it with embryonic stem cell, pluripotent stem cell, or you can use adult stem cell to do it. You can do it in vitro. That means most of the steps are done in test tubes. And later you inject the differentiated cell into patient. Or you can use drug to manipulate the stem cell. So everything happens inside the body. So there are many ways which are summarized on this slide. Okay, we are, I'm still trying to get used to this shorter format of class. I'll try to do 45 minutes in the future, but allow me to adapt. On the last slide, I show this data again. because today I have divided you guys to groups. Please check which group are you in, and also check if you belong, if I mistakenly put you into two groups, or I didn't put you into any groups. Let me know. I will fix that. And then please, hopefully during this weekend, before our next class, which is on next Monday, tell me which date your group will choose. Okay, you can choose 2 dates because every group will give a presentation twice. Okay, so each group will choose 2 dates. Tell me the dates you choose. That will be on a first come, first served base. If your chosen date has been taken by somebody else, I will let you know. I will arrange a different date for you. So because the first date, available date is 29th, which is a week from now, the group who unfortunately get assigned or pick that date, we can move that to the very last day of class, which is the 30th of April. Okay, we can make that happen if you don't want to do the presentation discussion next week. Okay, so please do check that out. Okay, that's the end of today's lecture.
Wait, where do we check to see our groups? Is it in Teams or is it on Blackboard?
Teams.
Do I go to, is it under post, class notebook, assignment, or?
It's a channel. It's a
Yes,
channel.
channel.
Oh, it's a channel. Okay, thank you. I see it. I'm over here in group 4. Thank you.
Okay.
Oh, Professor, so for those groups, it doesn't tell you like who your members are. How would you be able to find that?
Oh, you won't be able to tell who the other members are?
Yeah,
No,
so I don't know what's the communicate.
No, I can't see them.
And it will show the members.
Oh, it shows your members?
Yeah.
Oh, that's
Can someone
good.
explain how to do that, please?
Yeah, so just go on the top of your channel. Like for instance, if you're group 1 or 2, go to the top and then you'll find I next to the meet button.
Uh-huh.
And then click on I and you'll see all the members.
Oh, I see it.
Okay.
Yeah.
Oh, and then Professor, so for picking the dates we choose, I'm assuming we can't pick the same topic twice, right? So for example, like stem cells in adult animal tissues, we can't pick that twice.
That's true. I would prefer you to pick dates that's far apart from each other, not consecutive weeks.
Okay, thank you.
How do we get you the dates that we want to do? Like as a group, do we just put it in the conversation? Do we email it to you, or?
That's a good question. We can have that in next class.
Okay, sounds good. I like that.
Yeah, let's do it in class.
Okay, awesome.
right.
Can someone please tell me how to find the group? I'm still kind of missing on it.
It's channels in Teams.
Oh, Teams channels.
Channels, yes.
Okay.
If you go to Teams and do you see? Let me see.
How do we access the channels?